it is remarkable to me how our immune systems evolved. And they have this really brutal task, which is how can they be tuned to detect any foreign pathogen that is harmful without knowing a priori what that could be, while at the same, so in other words, how can you tune a system to be so aggressive that it can eradicate any virus or bacteria billions of years into the future without knowing what it's going to be? But at the same time, it has to be so forgiving of the self that it doesn't turn around and attack the self. It's remarkable. And of course, we can always think of the exceptions. There are things called autoimmune conditions. So clearly the system fails, and the immune system turns around and attacks the self. If you see a person with vitiligo, I have a little bit of vitiligo on my back, a couple of spots. Clearly the immune system is attacking something there and destroying some of the pigment.
I didn't realize vitiligo was autoimmune.
Yeah. So there are lots of, more serious autoimmune conditions, of course, somebody that has lupus or, where the immune system can be attacking the kidney, the immune system can be attacking any, autoimmune conditions can be deadly. But fortunately, they are very rare. And for the most part, this immune system works remarkably well. So how does it work? And why is it that cancer seems to evade it virtually all of the time? This is the question. Now, let's first of all talk about how it works. And then when I tell you how it works, you'll say, that sounds amazing. Clearly, it should be able to destroy cancer. I'm going to simplify it by only talking about one system, which is how T cells recognize and get activated, how T cells recognize antigens. So we have something called an antigen. So an antigen is an antibody generating peptide. So it's a protein, almost always a protein. They can be carbohydrates, but they're almost always proteins, and they're very, very small peptides. Like we're talking as little as 9 amino acids, maybe up to 20 amino acids. So teeny tiny little peptides. But it's amazing that in such a short peptide, the body can recognize if that's Andrew or not Andrew. I mean, when again, think about like, we talk about proteins in kilodaltons, right? We're talking about proteins in terms of thousands of amino acids that make up every protein in your body. And yet, if it samples a protein and sees that, hey, this little 9, 10, 15 peptide amino acid is not part of you, I know it's bad, and therefore I'm going to generate an immune response to it. So we have what are called antigen-presenting cells. You have cells that go around sampling peptides, and they will, on these things called MHC class receptors, bring the peptide up to the surface and serve it up to the T cell. There are two types of these. There's MHC class 1 and MHC class 2.
This is major histocompatibility complex.
That's correct. And we refer to them that way because because of the context in which they were discovered, which was for organ rejection. So not surprisingly, when you need to put a kidney into another person, if that kidney is deemed foreign, it will not last long. And the early days of organ transplantation were rife with immediate rejections. And by the immediates are the ABO incompatibilities, but the sort of next layer of incompatibility was MHC incompatibility, which would lead to, within weeks, the organ is gone, as opposed to within hours. So you have these two classes of MHC. You have class 1 and class 2. Class 1 is what we call endogenous. So this is basically what happens when a protein or an antigen is coming from inside the cell. So let's consider the flu. So if you get the flu, the influenza virus infects the respiratory epithelium of your larynx. And that virus, as folks listening might remember from our days of talking about COVID, viruses can't replicate on their own. What they do is they hijack the replication machinery of the host, and they use that either to insert their RNA or DNA to replicate. And in the process, proteins are being made. Well, those proteins are the proteins of the virus, not of us. So some of those peptides get launched onto these MHC class 1 glove, basically. The glove comes up to the surface, and a T cell comes along. And in the case of MHC class 1, it's a CD8 T cell. These are what are called the killer T cells, right? And so this cell comes along, and with its T cell receptor, the T cell receptor meets the MHC class 1 receptor with the antigen in it. And if that's a lock, it realizes that's my target. And it begins to replicate and proliferate and target those. And that creates the immune response. And by the way, that's how it works when you vaccinate somebody. You're basically pre-building that thing up.
So would this fall under the adaptive immune response or the innate immune response?
So this is adaptive. Yep. Innate is just the pure antibody response on the B cell side. I won't get into that for the purpose of this discussion. The other example is MHC class 2, and that's also part of the adaptive system, or the innate system, which is more what we call the exogenous form. So these are peptides that are usually coming from outside the cell. So we're going to focus more on the MHC class 1 because this is peptides that come from inside the cell. Okay, so just keep in the back of your mind, if a foreign protein gets presented from inside a cell to outside a cell, the T cells recognize that and they will mount a foreign response. And by the way, that's why we basically can beat any virus. Like, if you consider how many viruses are around us, the fact that we almost never die from a viral infection is a remarkable achievement of how well this immune system works.
We're constantly combating these viruses.
Constantly. And by the way, we don't really have very effective antiviral agents. It's not like antibiotics. Like, we have antibiotics up the wazoo. I mean, we're way better at fighting viruses than bacteria.